Welcome, everyone, to this session of our May 2025 MicroCap Conference. I'm Alex Handman, and I serve as an equity research analyst here at Sidoti & Company. Today, we're pleased to be in conversation with CEO Robert Bitterman of Phio Pharmaceuticals, ticker PHIO. During the presentation, please feel welcome to submit questions using the Zoom Q&A interface at the bottom of your screen. After the presentation, we'll open to your questions. And with that, Robert, I'll turn it over to you.
Thank you very much, Alex, and thank you everyone for tuning into the Phio story today. Phio is a clinical stage biopharmaceutical company that is currently specializing in the treatment of various types of cancer cells. And with that, we do this with our patented unique technology called INTASYL, which is a short interfering RNA that injects the tumor directly to help silence excess production of genes and also awaken in the process the T cells to effectively go and kill tumor cells. Our objective as an organization is to take our lead compound through the regulatory pathway to commercialization and to provide a better treatment modality in terms of efficacy, safety, tolerability, and convenience, and hopefully as well pharmacoeconomics. So with that, let me provide the forward-looking statements just since we will be talking about some things in the future. And with that, I will go directly to the core of the presentation here, and to start with saying that the Phio history has one of innovation. It was co-founded by a gentleman named Craig Mello, who actually was awarded the Nobel Prize for his discovery of RNA interference. That invention eventually led to the production of INTASYL, which is a short interfering RNA and is technically a synthetic drug, which has the ability to precisely silence genes in the human genome, and in so doing, making cells more effective in killing tumor cells. So the question may be, why do we want to silence certain genes? And the answer rests in the fact that certain elements in our DNA and messaging get out of balance, and there is excess production of proteins from certain genes that ultimately can lead to the proliferation of tumor cells. And what our technology does is silences specific genes that have that excess production protein production, and then it reactivates the body's natural immunity in the form of T cells, making those immune cells more effective in killing tumors. And it does so through a very precise self-delivering technology, which is the INTASYL. The INTASYL's value proposition really is designed to provide a safety, efficacy, and convenience that is very different than existing treatment modalities. And it also provides a versatility through this precision self-delivery. And all of this is protected by an extensive array of various patents. With regard to these value drivers, with regard to safety, we are in a phase 1b clinical trial at the moment, and through this stage of the study, there have been no dose-limiting toxicities or serious AEs in the study. Our drug, since it is injected directly into the tumor, minimizes the risk of off-target toxicities, and unlike monoclonal antibodies, which are systemically infused into the body, we do not have the impact of autoimmune serious adverse events, which are common with the antibodies. Our drug is also delivered in nothing other than buffered sterile saline solution. So it minimizes the potential toxicities of other drugs that use formulation enhancers or manipulations to get it to where it has to go. And finally, our drug is temporary in its effect. It is not permanent, such as genetic alteration. With regard to efficacy, through the study to date, 10 patients, 1 with melanoma, 9 with cutaneous squamous cell. In cutaneous squamous cell, 4 of those patients have had 100% tumor pathology clearance, 1 greater than 90%, 1 greater than 50%, and 4 patients are deemed as non-responders, but none of the patients that did not respond exhibit any progression of the disease. And finally, from a standpoint of convenience, this drug, because it's injected into the tumor, can be administered in the doctor's office, which avoids having to deal with infusion centers, which are required for monoclonal antibodies. The drug is also packaged in a way that can be individualized for each patient based on the size of the lesion. And if we're talking about the standard of care today, which is Mohs surgery, this sort of treatment in shrinking the tumor will minimize aftercare and in the surgery, such as plastic surgery, reconstruction. And finally, manufacturing of this drug is basic, simple chemical synthesis, which yields a relatively low cost of goods sold and can help in designing pricing for formularies in the future. So with regard to versatility, and this is where the interesting element of the self-delivering technology comes in, the drug can be delivered to any gene with specificity. It can be delivered to any cell type without formulation enhancers. It can even target multiple genes simultaneously, or it can be used in combination with other therapies as well. And this is a quick picture of what the chemistry looks like. It's an asymmetric duplex that consists of about 25 nucleotides, which are really synthetic fragments of RNA. And in this structure, there are three important things that basically make the product work, as well as enable the patent protection, which is very extensive. It has a cholesterol element, which allows the drug to be transferred successfully through the cell membrane intact to where it has to be deposited. There are phosphorothioates that basically protect the molecule from destabilizing elements in the body, these nucleases which break it down. But then finally, probably the most interesting is that we have the ability for exceptional gene target specificity based on how we design the target of this. And we can basically change the design depending on what gene we want to silence. Having said that, the last element of the value proposition is in the intellectual property. There are 76 patents issued and another 27 pending, and these patents cover a number of factors from the chemistry structure of the technology to specific compounds that we've been able to silence, as well as gene targets and therapeutic indications as well. So I'd like to shift now to strategy and just briefly mention that there's really two elements of it. One is what we call our self-directed clinical development, where we've taken two compounds that we are trying to take through the regulatory pathway. The second element of our strategy is to out-license a number of other compounds which are non-strategic to us, and I'll give you an example of that. Initially, our company was a discovery research company, and in that role, the scientists basically came up with a number of different compounds that they could silence through manipulating the INTASYL technology. We looked at this and said, a couple of years ago, we're going to have to find a compound that we can basically apply into man that can be effective medically and also at the same time can provide a market size that can provide a good return on investment. So we had to select where we could basically, given our limited bandwidth of personnel and cash, where are we going to put our efforts? And we use this algorithm in terms of determining what that would be. And it's essentially centered on risk mitigation. Which of these compounds and where could we place them where we would mitigate the risk or have the greatest likelihood of success? And looking at that in the context of both cost as well as lead time. And then if we could satisfy those three variables, look at the market potential of where this particular target would be, and also what the competitive landscape would be. And that would become then the basis for filing an investigational new drug with FDA. We ended up determining that our lead program, would be the silencing of the PD-1 gene with our intratumoral PH762 drug. The risk mitigation was really quite clear because PD-1 was implicated in skin cancer, and that was proven in the monoclonal antibody drugs from Merck and Regeneron. And we also knew that from our preclinical studies, we could silence PD-1 at its source directly in the T cell. And so with that, we then looked at the categories of where we could target this. And cutaneous carcinomas were one of the biggest and largest growing markets. And further, we looked at cutaneous squamous cell, which is the second largest solid tumor in the human body. And with that, we further looked at the number of people in clinical trials, and we found that there were really a limited number of trials going on with major players. So this became a very attractive target. Here you see the market size of the incidence of solid tumors with cutaneous squamous cell, CSCC, representing over 50%, 51% of those different types of tumors in the body. The second program was to silence the BRD4 gene with intratumoral 894. 894 was implicated in numerous cancers. It has the selective ability to go after the BRD4 gene alone, which is something that previously small molecule therapy studied by large pharma could not do, resulting in a number of serious adverse events. But in our case, the 894 targeting BRD4 was able to develop a clean toxicology profile in non-human primates, which gave us real indication that this could be a unique drug. Finally, 894 had the dual mechanism of action to go after not only killing the tumor, but also activating the immune cells at the same time. So this was an attractive follow-on program for us. And this ended up being our pipeline for drugs self-directed development. 762, which is currently midway through the phase 1b trial, and 894, which is now essentially IND ready with all the enabling studies ready to file an IND. So getting back to 762, we filed the IND, and the FDA gave us clearance within a month to study late stages melanoma, Merkel cell, and cutaneous squamous cell. But they gave us one additional recommendation, which was really important. And that was to be able to study both stages 1 and 2 in cutaneous squamous cell. And this was unique because it represented a treatment alternative for patients, and it also drove our economic model to get a return on investment. And the reason for this is that in stages 1 and 2, that represents over 70% of the incidences of squamous cell, and there are 1.4 million incidents in this area. And currently, and most importantly, no FDA drug has been approved at this stage for stages 1 and 2. The alternative, or the standard of care, is really surgical intervention in the form of Mohs surgery. And when we looked at this market alone, we looked at the addressable market for stages 1 and 2 as exceeding $20 billion in potential revenue. So here's a picture of cutaneous squamous cell in the facial area. The tumor size, the location, patient's general health is always going to present an argument for a non-surgical alternative. And in this case, when we looked at the positioning of 762, we essentially said this is a tissue-sparing drug. We can shrink, if not eliminate, the tumor. In so doing, we can minimize surgical excision and reconstructive surgery. Additionally, it provides for convenience because we can intratumoral injection can be done in the physician's office, unlike the monoclonals, which have to go to an infusion center. And finally, from the standpoint of toleration, the autoimmune adverse events associated with monoclonals are not existent to any significant extent with the 762 product because they're not systemically infused. And one last point, one of the things that we're learning is that T-cell recognition through our INTASYL program may actually result in the ability to prevent tumor recurrence, which is something that's very interesting and something that we're further looking into. Talking about the clinical trial, the purpose of the trial is to determine the dose for the next trial from a standpoint of safety and tumor pathology. And the trial currently has up to 30 patients in escalating dose concentrations. It represents 4 intratumoral injections over 3 weeks with a resection of any residual lesion at the end of week 5, looking for safety, pathology, tumor results, pharmacokinetics, and biomarker analysis. We have some quality sites that are basically enrolling these patients, and we expect that the total inpatient enrollment will be completed by the end of the third quarter of 2025. And so I give you here a picture of the adoption curve, the technology adoption curve relative to the existing treatment modalities for skin cancer. And you can see with this that we, as marked by the star on the left, are at the very early stages of technology adoption because this is the first product of its nature in immuno-oncology to actually to be in man at this point. On the other extreme, of course, we have the cryosurgery, which everyone uses in the derm world, and Mohs surgery, which is very well accepted. But the goal here is as we get more information and knowledge about the success of our trial, that this will move up the adoption curve and start to become much more acceptable and aware among a much larger portion of the total population that could be interested in using this type of therapy. So, a couple of final points here. We need to talk about not just our own development, but there, as you saw from the prior slide, that we have a number of other compounds. And the question is, how can we return or get the maximum return on the rest of our portfolio through business development? And there are some opportunities for collaboration in a number of places. First of all, 762 has other potential indications other than skin cancers. If we can alter the genetic or the sequence of the nucleotides in the chemistry, because anything that's subject to gene silencing could be a potential target. We also have demonstrated in man that in the cell therapy, which is called adoptive cell therapy, that our 762 enhanced a certain type of T cell and was showing to remove a significant portion of lesions in a late-stage melanoma patient, although that was a short-lived trial. We also have the ability to increase the potency and yield of natural killer cells with 762. Uh, Other areas of potential collaboration are to use in combination with monoclonals because our product works to silence PD-1 inside the T cell, where monoclonals work on the cell surface. So in combination, we were able to demonstrate preclinically that using them together could actually enhance tumor suppression. And then finally, some of our compounds actually have dermatological application because they can basically contribute to the reduction of hyperpigmentation and photoaging. So the ability to capitalize on those through business development provides another mechanism for us. I'll summarize now with metrics and some leadership. Today, at the end of March, we had approximately $13 million in cash on hand. We burn at a quarterly rate of about 2 million. We currently have headcount of 5 and an additional subject matter experts who are contracted individuals that we've worked with in the past who help complement our headcount. Our shares outstanding are about 4.8 million. We have 6.6 million in warrants, and those warrants are exercisable between the price range of $2 and $3. And if fully exercisable, that would be worth an additional $16 million in cash to us. Currently, our stock, I believe, closed yesterday at $1.78 or $1.79, and we have no debt on our balance sheet. And with respect to our leadership team, I'm working with a team of people who I've worked with over a number of years in my prior companies. We have great familiarity with each other, and they have very successful experience in directing previously 6 new drug applications and 1 Class 3 medical device approval through FDA. I've had experience in 8 commercial dermatological drug launches as well in the marketplace. And so with that, I will summarize and say that Phio Pharmaceuticals addressing a potential market of $20 billion with a very unique self-delivering technology and an extensive array of intellectual property and working through a leadership group that has had a track record of success in getting drugs approved. And with that, I leave you with making immune cells more effective in killing tumor cells, not just with 762 today, but potentially with a number of other applications of our technology as well. And so with that, I will turn it over to Alex to take your questions.
Robert, thank you so much for sharing a lot of interesting and exciting information. I'll remind folks if they have questions, they can submit them using the Zoom Q&A interface at the bottom of the screen. But maybe we can start with what you just mentioned, which is you're in the rare camp of having both clinical stage assets and proprietary delivery. And I think you've shown clearly the skincare market is large and you have the data for it. But maybe you could talk a little bit more about the platform potential beyond skincare and maybe even beyond oncology, which you mentioned briefly.
Sure. Well, the technology, if you remember that brief picture I showed of the asymmetric duplex, if we alter the sequence of those nucleotides, we can target other genes. And some of those other genes can affect other types of cancer from uh, liver cancer to lung to breast and cervical as well as prostate. So all of those are potential applications. And that's just with 762 and 894. There's also the possibility of being able to have an anti-inflammatory effect in certain areas as well. So those are areas, I mean, I have to say that our team actually has many years of experience in the dermatological arena, and that's kind of why we gravitated to that, sticking to our knitting, if you will. It's best to do the things that you know well. But for other companies in other areas, being aware of the ability of this technology and its intellectual property protection, there's people specializing in other forms of cancer. This could be very effective and useful for them.
Great context. Thank you very much. And you're one of the few small caps, with an in-house RNAi delivery platform. So we're getting some questions from the audience. How do you think about building value internally versus, maybe partnering externally?
Well, we're open to partnering on a number of different fronts. But I showed you a brief snapshot of the technology adoption curve, which is something that's been tried and true over decades. And the reality is that most big companies, they are not early adopters. In fact, they are the late majority. In fact, Big Pharma was described to me by one of the largest CROs as being the last to be first and the first to be last. If it's not invented in their house, they're not going to have that much interest in it because they don't trust what they don't make themselves. So to partner successfully with large companies, we're probably going to have further intellectual, or not intellectual, but further clinical results that move beyond the phase 1b trial and go into further studies that hopefully will be a phase 2b/3 based on the results that we're getting so far on safety and efficacy. But we're certainly open to that. But in the meantime, we have to slug through with our existing team to move and complete this trial and the other elements that go along with being able to go to FDA to get the direction on the next stage. But we have reached out to a number of companies, and many of them said, you're still too early for us. And some early stage companies that were really too early for us that really couldn't provide us incremental value.
That makes sense. And I think that brings us to another question, which is, could you help us connect the dots from where you are now to the milestones you just mentioned and also help us understand how your current, cash balance aligns with some of those key upcoming milestones?
Yeah, sure, I'd be happy to. So we mentioned this is a dose escalation trial with three cohorts completed. The fourth, all of the patients in the fourth cohort are in treatment phase. And so with a 5-week window in order to get through determining safety, you can do the math and figure out that we'll be reporting results on safety in fairly short order. Uh, but after that, there's ideally one more cohort to go, and that at that point, the fifth cohort, if we go directly to number 5, that will be the maximum amount of physically possible concentration of dose that we can actually put in the drug. With the results of that, simultaneously we are working on our CMC area to qualify a commercially scalable supplier of active pharmaceutical ingredient. And we expect that we will be signing an agreement with a US-based organization within the month. We're fairly down the path with that. So we'll have to basically produce product API material to make the next round of clinical trials, which would then be viable for commercial use. And some of that drug will also have to be put into a one further study that the FDA has asked us to do on a marmoset study, a 3-month marmoset study as well, just to give them additional assurance on the safety. So those are the steps to get through all the elements of the phase 1b trial, along with chemistry and tox. That will take us somewhere toward the middle of next year with everything completed. And to get to that level, we will have probably the need to raise, in the absence of warrants cashing out at the $2 to $3 level, we would probably need another 10 to $12 million of cash to get to that point in the absence of the warrants cashing. And at that point, we would then be looking to set up a phase 2b/3 trial, FDA willing. But based on the safety and the efficacy we're seeing today, we kind of are optimistic that that might be where the FDA would let us go. And then doing a phase 3 type trial or a pivotal trial, 2b/3, is going to be a function of the number of patients that the FDA requires in that trial. And that could be that you never know. But if we pick a number, an estimate of, say, 250 for rough numbers, and I'm not saying that that's exactly the number, and pick a number of $100,000 a patient, which I think would be on the high side. We'd be talking is somewhere around $25 million to complete that before we could file an NDA. But I think that's looking out, it's kind of on the conservative side, but that's what's in my economic models today.
Great context. Thank you. And maybe one last question, given trends in the stock price over the last year, are there particular aspects of the business that the street may be underappreciating, for folks obviously who are familiar with the name, and then for folks who are new to the name, if you could sort of combine that with summing up the value proposition, if they might be looking across, gene therapy investment opportunities.
Well, I think the real value in this program, notwithstanding what we've seen to date in the phase 2 or 1b trial, is the potential to target a sector of skin cancer which technically has no approved alternative other than surgical intervention. And with that, if we look at the market, the addressable market of $20 billion, a modest share, pick a number, 5%, and pick a modest pricing for the regimen. I'd say 15 million or $15,000 in today's dollars, which is a fraction of what the monoclonals charge for their drugs, would easily get you to $1 billion potential just with a 5% share and a very, very modest pricing compared to what's in the market today. And our cost of goods also is, we know what it costs from the bottom up, and that will provide for very attractive margins while at very conservative prices, so well in excess of 90%. So that gives us flexibility with formulary pricing. But aside from that, this technology is extremely versatile, and it can be applied to a number of different indications. And I think as more and more people get comfortable with the results of this trial, where we have, the ability to get 100% pathological clearance in that number of patients in a matter of 5 weeks is it's been surprising to us and also surprising to our clinical investigators as well. And so therein, I think, lies the upside potential to realize that we can basically do this with a reasonably inexpensive burn because of our lean overhead. And with that also, the amount of cash that we would have to raise going forward on some attractive inflection points through the next cohorts probably gets us to where we have to go and keeps the stock moving in the right direction.
Great context. Yeah, thank you for walking us through. And we are at time, so I would like to, pause here and thank you, Robert, for sharing the Phio Pharma story with us, and also thank everybody listening for spending time with us today.
Okay. Well, thank you very much. And keep in mind that this is Skin Cancer Awareness Month. And I'd recommend that everybody get a skin check at least once a year with a dermatologist because you don't want to let these things go in the wrong direction.